Imagine a microscopic force, an unseen inhabitant that has journeyed with humanity for, well, millennia, silently shaping our health, our vulnerabilities, and even surprisingly, our protections. It's a compelling thought, isn't it? Today, we're embarking on a deep dive into one such ancient co-colonizer, a bacterium that still impacts an astonishing half of the world's population, Helicobacter pylori, or H. pylori. Our mission in this deep dive is to truly unpack everything about this remarkable microscopic inhabitant of our stomachs. We're going to move beyond just the headlines and explore its intricate biology, its surprising dual roles in both disease and protection, how we detect it, and the very latest strategies clinicians use to manage its presence. This isn't just about accumulating facts, it's about understanding and really mastering this information with the depth and nuance expected of, well, the elite professionals in this field. And guiding our journey today is a comprehensive chapter from a leading medical text, specifically Chapter 163, which provides a rich foundation for everything we're about to discuss.
That's right. And to begin our mastery, let's first get to grips with what H. pylori truly is. At its core, H. pylori is a gram-negative bacillus. Now, if you're picturing something maybe a bit alien, you're not far off. Imagine a tiny, rod-shaped bacterium with a specific type of cell wall that helps us classify it. What's truly distinctive about H. pylori, though, is its spiral shape.
Spiral. That sounds unusual.
It is. And it isn't just for looks. This spiral morphology, coupled with several whip-like tails called flagella, gives it remarkable motility. It allows it to literally corkscrew its way through the very viscous environment of the gastric mucus in your stomach.
Ah, yeah.
It's like a tiny drill bit, expertly navigating a thick, gooey landscape.
So it's built for speed and precision within that specific environment. But our stomachs, they're notoriously acidic, aren't they? Like incredibly harsh. How on earth does a bacterium survive that?
Yeah, you've hit on its most remarkable survival strategy. H. pylori has evolved several sophisticated mechanisms to thrive in that incredibly harsh, acidic stomach environment.
Yeah.
The most notable of these involves a highly expressed enzyme called urease. Think of this urease enzyme as a tiny chemical factory, right?
Okay.
It takes urea, a compound naturally present in your stomach, and it breaks it down into ammonia and carbon dioxide. The ammonia specifically then acts as a powerful buffer.
Buffering the acid.
Exactly. Neutralizing the surrounding gastric acid and creating a more hospitable, sort of pH-balanced microenvironment for the bacterium to live in. This acid resistance mechanism is, well, absolutely crucial for its long-term persistence in a place that's designed to break down almost everything else.
So it's essentially creating its own little pH balanced sanctuary right there in the stomach lining. That's incredibly clever, almost like it's engineered its own survival kit.
It really is quite ingenious.
And where exactly does it set up shop then? Is it just floating around in the stomach acid, or does it attach somewhere specific?
Good question. It primarily lives within the gastric mucus layer, that protective slime coating your stomach lining. While a significant proportion of the bacteria adhere directly to the mucosal surface, essentially sticking to the wall beneath the mucus. It's also been observed that a very small number, a tiny fraction really, may potentially enter the epithelial cells themselves or even penetrate deeper into the mucosa. But it's important to understand that its distribution is overwhelmingly localized to that stomach lining and mucus. It's not typically spreading systemically throughout the body.
Right.
In terms of its growth requirements, H. pylori is microorophylic, meaning it prefers and thrives in low levels of oxygen, not completely oxygen-free like some bacteria, but definitely not high oxygen like the air we breathe. It's also a slow-growing organism and requires quite complex growth media when we try to culture it in vitro, in the lab. That makes it somewhat challenging to study effectively sometimes.
What's truly astonishing, though, is the sheer antiquity of its relationship with us. I mean, we're not talking about a recent invader here or some new bug that just popped up recently.
No, not yet.
This bacterium has naturally colonized humans for at least 100,000 years, and likely throughout the entirety of human evolution. Think about that for a moment. It's been with us essentially since our very earliest ancestors.
It's staggering, isn't it?
It really is. This isn't just a random pathogen. It suggests a deeply intimate and long-standing co-evolutionary relationship. Does that imply it's not always just a pathogen? Does such a long relationship mean it might have some, ingrained role, even if we're only just discovering it?
It absolutely does, and we'll definitely get to those surprising roles later on. But this long history is certainly reflected in its global prevalence today. When we look at a current snapshot, Roughly 50% of the world's human population is colonized by H. pylori.
Half the planet. Wow.
Yeah, it's huge. And once acquired, this colonization often persists for life unless it's specifically eradicated through antibiotic treatment. There's also a stark contrast in prevalence rates across different regions.
Huh.
In most developed countries, think the US, Europe, Oceania, the prevalence among adults is less than 30%.
Okay.
However, in many developing regions, places like parts of Africa, South America, and West Asia, this figure jumps significantly, often over 60%.
That's a massive difference. Makes you wonder about the factors driving that disparity. Does age play a role? Are younger generations less likely to have it these days?
You're touching on a key point there. Age is a significant factor, yes, but it's often misunderstood. In the United States, for example, the prevalence can be as high as 50% in 60-year-old individuals. Yet it's only around 20% in 30-year-olds and remarkably less than 10% in children.
So it increases with age.
Well, not exactly. This isn't primarily because people acquire it more as they age. Instead, it's largely due to what we call the birth cohort effect.
Birth cohort effect.
Yeah. What this means is that current 60-year-olds were simply much more commonly colonized as children than today's children are. H. pylori is typically acquired in childhood, you see. Spontaneous acquisition or loss of H. pylori in adulthood is actually quite uncommon.
Ah, I see.
So the main risk factors for infection tend to be markers of, well, crowding and social deprivation during childhood, which makes sense in the context of its usual acquisition route.
Okay.
Over the past half-century, longitudinal studies have consistently shown a declining prevalence of H. pylori, which correlates pretty well with both socioeconomic development and, of course, the widespread use of antibacterial treatments.
That's fascinating. So it's not that older people acquire it more often, but that they grew up in a time and environment where it was simply much more common.
Exactly.
Does that mean as our societies continue to develop, better sanitation, less crowding maybe, we might actually see H. pylori become even rarer, potentially changing the whole landscape of these stomach diseases entirely?
That's precisely the long-term trend we seem to be observing, yes. If it's primarily acquired in childhood, the next question becomes, how does it actually spread? Are we talking about, a handshake, a cough, or something else?
Right. How does it pass from person to person?
Well, the first crucial point is that humans are the only important reservoir for H. pylori. You're not generally going to pick it up from a pet or some environmental source in the same way you might with other bacteria.
Okay, so it's human to human.
Predominantly, yes. Children usually acquire the organism from their parents, most often the primary caregiver, particularly in developed countries. In less developed countries, acquisition from other children can be more common, perhaps due to closer living conditions or play environments. Now, regarding the specific transmission routes, there's still some ongoing uncertainty and, frankly, debate about whether it's more often fecal oral or oral oral.
Fecal oral versus oral oral. What's the evidence?
Well, the evidence we have suggests it's easily cultured from vomitus and gastroesophageal refluxate, which definitely points towards an oral component, maybe sharing utensils, drinks, food, that sort of thing. However, it's much less easily cultured from stool samples, which might seem to contradict the fecal oral hypothesis, but it doesn't entirely rule it out. Contamination of food or water is still a possibility, especially in areas with poor sanitation. What is clear, though, is that most acquisition happens during those early years of childhood, often within families. And while our deep dive today is really focused on H. pylori, it's worth briefly mentioning that it's not alone. There are other Helicobacter species out there, part of a diverse family beyond just pylori. Yeah, a small proportion of what we call gastric Helicobacter infections are actually due to these other species. These non-pylori gastric Helicobacters are possibly acquired from animals we call that zoonotic transmission, and they're typically associated with sort of low-level inflammation, though they can occasionally cause minor disease. Furthermore, in individuals with compromised immune systems, several non-gastric or intestinal Helicobacter species can cause an illness with clinical features that resemble those of Campylobacter infections, which is a different family of bacteria entirely. So it just highlights that the Helicobacter genus is diverse and can impact human health in various ways beyond just our main subject today, H. pylori.
That's a fascinating overview of this ancient and, well, incredibly widespread microbe. We've seen how it's built to survive and how it spreads. But it's not just a benign passenger, is it? You mentioned it seems to be a real double-edged sword.
That's a good way to put it.
And while it's incredibly prevalent, only a fraction of those colonized actually develop overt disease. But one thing is almost universal, right? You said essentially all H. pylori colonized persons develop what we call histologic gastritis.
Yeah.
What exactly is that, and what's happening in our stomachs when that occurs?
Yes, that's a critical point. Histologic gastritis basically means if you look at the stomach tissue under a microscope, you see signs of ongoing low-level inflammation. It's characterized by certain immune cells. Think of them as tiny foot soldiers of your immune system moving into the stomach lining, setting up a persistent kind of battle there. Now, even though H. pylori has evolved numerous adaptations to avoid, shall we say, overstimulating your immune system, and even though it has coexisted with humans for so long, its colonization still provokes a significant and persistent local and systemic immune response. This includes the production of antibodies and cell-mediated responses.
But you said it persists for life often, so the immune response doesn't clear it.
Exactly. Here's the paradox. These immune responses are remarkably ineffective at actually clearing the bacterium. This inefficiency appears to be partly due to H. pylori's own sophisticated mechanisms to downregulate or sort of suppress the host's immune system. This ultimately fosters its long-term persistence in the stomach. So you have this ongoing inflammatory battle, but one that your immune system seems to be losing, allowing the bacterium to stay put indefinitely in many cases.
That leads us to the million-dollar question then, doesn't it? If essentially everyone who's colonized develops this gastritis, this background inflammation, why do only a relatively small percentage, you said around 10 to 15%, actually go on to develop overt disease like peptic ulcers or even gastric cancer? What differentiates those who get sick from those who don't? What's the secret sauce that pushes some into serious illness while others remain largely unaffected?
That's precisely where the complexity lies, and where mastery of this topic really comes in. It's not a simple one-to-one relationship like infection equals disease. It's much more nuanced. It's really a tango, a dynamic interplay between three main factors: differences in the bacterial strain itself, the host's own genetic makeup, and various environmental factors.
Okay, strain, host, environment.
Exactly. Understanding this combination is absolutely key to truly understanding H. pylori disease and anticipating clinical outcomes. Let's delve into those bacterial strain differences first. These are often referred to as virulence factors, basically because some strains are simply more aggressive, more virulent than others. One of the most studied and frankly fascinating aspects is something called the CAG island.
The CAG island. Sounds intriguing.
It is. This isn't just a single gene, it's a whole cluster of genes, a specific region on the bacterial chromosome. And together, these genes encode a bacterial type IV secretion system.
Type IV secretion system? What does that do?
Imagine it like a tiny microscopic syringe that the bacterium literally uses to inject its own proteins directly into our stomach epithelial cells. That's quite an advanced mechanism.
Wow, like a direct injection.
Precisely. Through this ingenious system, a key effector protein called CAG is injected into the host cells. Once inside, CAG gets a molecular on switch, flipped a process called phosphorylation, and this activated CAG then sets off a cascade, a whole chain reaction of host cell signaling.
And the result of that signaling?
The result is profound changes within the cell. It alters its growth, its structure, its cytoskeleton, and importantly, its inflammatory responses. Essentially, H. pylori is actively manipulating our own cells from the inside, forcing them to behave differently, which then fuels the disease process. It's not just sitting there causing passive irritation. Another important component of this CAG system is CAG. This is a protein located at the very tip of that secretory apparatus, a syringe needle if you like. CAG plays a crucial role by binding to specific receptors called integrins on the surface of our host cells. Think of integrins as molecular handholds on the cell surface. This binding further amplifies the signaling pathways within the cell, intensifying the bacterial influence. And it doesn't stop there. Soluble components of the bacterial peptidoglycan cell wall, basically, fragments of the bacterial outer layer, also enter the host cell via this same tiny syringe system.
More stuff getting injected.
Yep. Once inside, these peptidoglycan components are recognized by an intercellular bacterial alarm receptor called NOD1. This recognition by NOD1 then stimulates a potent pro-inflammatory cytokine response, leading to an enhanced and more vigorous immune reaction from the host.
And the clinical relevance of all this CAG stuff?
It's incredibly significant. Carriage of CAG-positive strains of H. pylori strains that have this whole injection system is strongly associated with an increased risk of both peptic ulcer disease and gastric adenocarcinoma. It's a major marker for more severe outcomes. It's truly a masterclass in microbial manipulation, how it exploits our own cellular machinery.
So, if I'm understanding this correctly, it's not just causing inflammation passively by being there. It's actively reprogramming our own cells from within, forcing them to behave differently, which then fuels the disease. That's a level of cellular hijack I hadn't quite imagined from a bacterium.
That's a great way to put it, cellular hijacking.
What about other bacterial factors besides this CAG system?
Indeed, it's a sophisticated system. Another major host interaction factor produced by H. pylori is the vacuolating cytotoxin, VACA. Yes. This is a protein that forms pores, or tiny holes, in cell membranes, disrupting their integrity and leading to changes within the cells, including the formation of vacuoles, hence the name. VACA is also polymorphic, meaning there are different forms of it, like different versions of the same software, some more potent than others.
And like CAG.
Exactly. Just like with CAGCO, carriage of H. pylori strains that produce more active forms of VACA significantly increases the risk of both ulcer disease and gastric cancer. So having both a functional CAG system and a potent VACA toxin makes a strain particularly nasty. Beyond these major players, there are other bacterial factors that contribute to increased disease risk as well, such as adhesins. These are proteins in the bacterial surface that help it stick to our stomach cells. For example, BABA is an adhesin that helps the bacteria bind to specific blood group antigens on the surface of epithelial cells. These adhesins are critical for initial colonization and persistence, essentially helping the bacterium gain a secure foothold in that harsh environment. Okay, so that covers the bacterial side. Some strains are just nastier. Now let's shift to the host side of the equation. Why do some individuals respond differently, even to the same strain? You mentioned host genetics.
Right, our own DNA.
Precisely. The best characterized host determinants of disease are genetic polymorphisms, essentially. Tiny variations in our own DNA sequence that differ between individuals that lead to an enhanced activation of our innate immune response when faced with H. pylori. A prime example is variations in cytokine genes. Cytokines are signaling molecules of the immune system, so variations in the interleukin-1 gene, for instance, or in genes encoding bacterial recognition proteins like toll-like receptors, TLRs, can make a difference.
How so?
Well, for instance, colonized individuals who have specific variations in their interleukin-1 gene, which cause them to produce more of this particular inflammatory signaling protein in response to H. pylori infection, are known to be at an increased risk of developing gastric adenocarcinoma.
So their own immune response, trying to fight it, actually contributes to the cancer risk.
In a way, yes. Their body's immune response is sort of genetically programmed to overreact in a way that, over the long term, becomes detrimental and promotes the changes leading to cancer.
Yeah.
It's a complex interplay. Beyond our genes, environmental cofactors play a crucial role in pathogenesis too. They act like accelerators or decelerators for disease development in people who are colonized.
Like what?
Smoking, for example, is a big one. It significantly increases the risks of both duodenal ulcers and gastric cancer in individuals who are H. pylori positive. It's an additive risk. The combination is definitely worse than either factor alone. Diet is another key environmental factor. Diets that are high in salt and preserved foods, think smoked, pickled, or salted foods, have been linked to an increased risk of gastric cancer in colonized populations.
And protective factors.
Conversely, diets rich in antioxidants and vitamin C, typically found in abundance in fruits and vegetables, appear to offer a modest protective effect against the development of cancer in colonized individuals. So it really is this delicate balance of bacterial virulence factors, individual genetic susceptibility, and lifestyle choices like smoking and diet that ultimately determines who gets sick and with what.
That makes perfect sense. A really complex interplay of host, bug, and environment. But here's another critical point you touched on. The pattern of the gastritis itself seems to dictate which disease a person is more likely to develop. Can you elaborate on the distribution of gastritis and its differential disease risk? It's not just if you get gastritis, but where in the stomach it is.
Yeah,
right?
absolutely. This is a fundamental concept for understanding the clinical manifestations and mastering this topic. The specific pattern of the gastric tissue response, where the inflammation is most intense, is directly associated with the type of disease risk. For instance, antral predominant gastritis, meaning the inflammation is primarily located in the antrum, which is the lower part of the stomach, near the exit leading to the duodenum.
Okay.
That pattern is most closely linked with the development of duodenal ulceration, ulcers in the first part of the small intestine. In contrast, pangastritis, which involves inflammation throughout the entire stomach, pan means all, or corpus predominant gastritis, where the inflammation is more concentrated in the upper body or corpus of the stomach.
Right.
Those patterns are linked with gastric ulceration, ulcers in the stomach itself, and more importantly, with gastric adenocarcinoma or stomach cancer. This difference in inflammation patterns likely explains a crucial clinical insight. Why patients who develop duodenal ulceration are generally not at a high risk of developing gastric adenocarcinoma later in life, even though they are colonized by H. pylori.
Because the inflammation is in the wrong place for cancer.
Essentially, yes. Their inflammation pattern, being primarily in the antrum, steers them towards duodenal ulcers,
Yeah.
rather than the more widespread acid-reducing atrophic changes in the corpus that are typically associated with the pathway to stomach cancer. It's a classic example of how the location and pattern of the chronic irritation matters profoundly for the ultimate clinical outcome.
That's a really important distinction. So let's dig into the pathogenesis of these key diseases a bit more, starting with duodenal ulceration. How does H. pylori in the stomach actually cause an ulcer down in the duodenum, the first part of the small intestine?
Yeah, the pathway for duodenal ulceration is becoming increasingly clear, though still debated in some details. H. pylori colonization and the resulting tissue responses, primarily in the gastric antrum, that lower part of the stomach, lead to a decrease in the number of specific hormone-producing cells called somatostatin-producing D cells.
Somatostatin? What does it normally do?
Somatostatin normally acts like a brake. It inhibits the release of another hormone called gastrin, and gastrin is the main stimulant for acid production in the stomach.
Okay, so fewer D cells means less brake.
Precisely. With fewer D cells, the brakes are off, and gastrin levels in the blood become higher than in H. pylori negative individuals. These elevated gastrin levels then lead to increased meal-stimulated acid secretion from the relatively spared gastric corpus, the main body of the stomach, which isn't as heavily inflamed in this antral predominant pattern.
So more acid is being produced overall.
More acid, particularly after meals. Now, how this situation specifically increases duodenal ulcer risk is still debated, but the prevailing theory is that this increased acid load being dumped into the duodenum may contribute to the formation of what's called gastric metaplasia in the duodenal bulb.
Gastric metaplasia, stomach-like cells in the intestine.
Exactly. Patches of stomach-like cells appear in the duodenum, which is normally lined by intestinal-type cells. These metaplastic areas in the duodenum then become vulnerable. They can be colonized by H. pylori, which likes stomach-type cells, subsequently become inflamed, and eventually ulcerate, leading to the characteristic duodenal ulcer. It's like the stomach's environment, including its susceptibility to H. pylori, is sort of spreading downstream into the duodenum.
Fascinating. And what about gastric ulceration, an ulcer in the stomach itself, and gastric adenocarcinoma, stomach cancer? Are the mechanisms similar, or are they completely different beasts?
The pathogenesis of gastric ulceration and gastric adenocarcinoma is, in some ways, less well understood than duodenal ulcers, though both are clearly associated with those other patterns. Pangastritis, or corpus predominant gastritis. While the same hormonal changes we discussed, the altered gastrin and somatostatin levels can still occur, the tissue responses in these cases are more widespread or concentrated in the acid-producing gastric corpus. Gastric ulcers themselves often develop right at the junction of the antral lower stomach and corpus-type upper stomach mucosa. These tend to be areas of particularly intense inflammation.
And gastric cancer, that sounds like a longer process.
It generally is. Gastric cancer usually arises in stomachs that have undergone extensive atrophic gastritis, where the stomach lining has thinned out, wasted away, and lost its normal function, and often hypochlorhydria, meaning significantly reduced stomach acid production.
Why does that happen?
It's thought that this process stems from progressive DNA damage accumulating within the stomach lining cells over many years, and the survival and proliferation of abnormal epithelial cell clones. This DNA damage is believed to be caused principally by reactive oxygen and nitrogen species, nasty chemical byproducts generated by the chronic inflammatory cells, like neutrophils and macrophages. They're constantly fighting the H. pylori infection over decades.
So the inflammation itself damages the DNA.
That's the leading theory. There's even speculation that other bacteria, ones that normally wouldn't survive in an acidic stomach but can survive in a stomach with reduced acid, hypochlorhydria, might move in and contribute further to this damage and inflammation. For the most common type of gastric cancer, the intestinal type, longitudinal analyses studying patients over time involving gastric biopsy specimens taken years apart from the same patient have clearly shown a stepwise progression. It typically moves from simple chronic inflammation, gastritis, to gastric atrophy, thinning, then to intestinal metaplasia, stomach cells changing to look like intestinal cells, then to dysplasia, precancerous changes, and finally to invasive cancer. It's often called the chorea cascade. It's like a slow, somewhat predictable journey down a dangerous path. However, there's also a diffuse type of gastric adenocarcinoma, which is less common overall, but more frequently found in younger adults. This type seems to arise more directly from chronic gastritis without necessarily going through all those intermediate atrophic and metaplastic stages. It's a bit different pathologically.
And you mentioned a concerning trend recently.
Yes, a noteworthy and somewhat concerning trend that's emerged in recent years is a progressive rise in gastric cancers. They're specifically centered on the gastric corpus, the upper part of the stomach. These are occurring in younger adults, typically those under 50 years old.
Younger adults.
Yeah, and these cases are also disproportionately seen in females. Critically, they appear to be arising without any detectable involvement of H. pylori.
Without H. pylori.
Correct. This suggests there may be other yet to be fully understood factors driving this specific subset of gastric cancers, representing a distinct and rather worrying development that's separate from the classic H. pylori-driven pathway. Research is ongoing to figure out what's causing these.
That's definitely a trend to watch. Okay, let's move on to another malignancy linked to H. pylori, gastric mate lymphoma. How does H. pylori fit into that picture? The idea of a cancer responding dramatically to antibiotics still blows my mind a bit. What does that tell us about the nature of these particular lymphomas?
It's truly one of the most striking examples of H. pylori's impact on human health, and a fascinating biological story. Gastric malt lymphoma, MALT, stands for mucosa-associated lymphoid tissue. Lymphoma is a relatively rare type of low-grade B-cell lymphoma that arises in the stomach lining. Interestingly, before the discovery of H. pylori back in the early 80s, these tumors were reported at a much lower rate. Since H. pylori was identified, the reported rates have increased substantially. Now, this might partly reflect overdiagnosis or better recognition, as pathologists became more aware of looking for them once the link was known. Pathogenetically, these tumors arise from the chronic stimulation of specific immune cells, lymphocyte populations, specifically B cells, in the stomach lining by the persistent H. pylori colonization. The immune system, in its prolonged, decades-long fight against the bacterium, essentially gets stuck in overdrive, leading to this abnormal lymphoid proliferation that eventually becomes cancerous.
And the antibiotic response.
That's what's truly remarkable and clinically significant about these low-grade MALT lymphomas. Numerous reports, starting soon after H. pylori's discovery, have shown them responding dramatically to H. pylori eradication therapies. Many of these tumors can regress, either fully or partially, just by getting rid of the bacteria with antibiotics.
Incredible.
It's a powerful demonstration of how removing the chronic inflammatory stimulus, the H. pylori, can actually reverse the disease process, even when it's technically a cancer. However, there's an inherent uncertainty sometimes in distinguishing between a true malignancy, a monoclonal proliferation, and benign lymphoid hypertrophy, which is just an overgrowth of normal lymphoid tissue reacting to the infection, which can look like a tumor. Among those patients whose tumors respond completely to H. pylori eradication, many do not have a specific genetic signature, a characteristic chromosomal change, specifically a translocation known as T1118 that's often seen in true, more aggressive MALT malignancies. So these responsive cases may, in fact, sometimes be benign polyclonal lymphoid proliferations rather than true cancers.
Oh.
Interestingly, though, Kaga-positive H. pylori strains, those more virulent ones we talked about, have been significantly associated with gastric malt lymphoma cases that do have that specific T1118 chromosomal translocation compared to MALT cases without it. This again highlights the intricate link between specific bacterial virulence factors and the development of certain types of malignancy.
That's a truly powerful insight. A cancer that can often be cured just by treating the underlying bacterial infection. Now let's talk about the more common, perhaps less dramatic, but still very significant ways H. pylori makes itself known, the bad news, if you will, the bread and butter clinical problems.
Indeed. The most common and direct clinical manifestation we attribute to H. pylori is peptic ulcer disease. Worldwide, it's estimated that approximately 70% of duodenal ulcers and about 50% of gastric ulcers are directly related to H. pylori colonization.
So the majority of ulcers.
Historically, yes, and still globally. However, it's crucial to note a shifting landscape, particularly in developed countries. The proportion of gastric ulcers in the stomach, caused by aspirin and nonsteroidal anti-inflammatory drugs, or NSAIDs, is actually increasing. In some developed countries now, NSAIDs have overtaken H. pylori as the primary cause of gastric ulcers specifically.
Interesting shift. But the link between H. pylori and ulcers is still very strong.
Oh, absolutely. The evidence supporting H. pylori's role as an ulcer-promoting agent is incredibly strong and comes from multiple lines of systematic investigation. First, the presence of the organism is clearly a significant risk factor for developing an ulcer. Second, non-NSA-induced ulcers rarely develop in the absence of H. pylori. If you don't have H. pylori and you're not taking NSAIDs, your chance of getting an ulcer is very low. Third, and perhaps most dramatically, eradication of H. pylori infection virtually abolishes the long-term relapse of these ulcers. Before we knew about H. pylori, ulcers were a chronic relapsing condition. Now, treat the bug, and for most people, the ulcer problem is solved permanently. It's like hitting a reset button.
Right.
And fourth, experimental H. pylori infection of gerbils, which is a common animal model for studying this, can directly cause gastric ulceration, confirming its direct role in causing the damage. So while H. pylori is neither absolutely necessary, NSAIDs can cause ulcers too, nor completely sufficient on its own, not everyone with H. pylori gets an ulcer, for peptic ulcer disease to occur, it is undeniably a very, very strong risk factor, and its removal profoundly alters the natural history of the disease, often providing a permanent cure.
Okay, ulcers are clear. Beyond ulcers, what about the more severe outcomes like gastric adenocarcinoma and that malt lymphoma? Again, just to recap their status as bad news.
Right. For gastric adenocarcinoma and lymphoma, prospective studies following large groups of people over time have consistently shown that H. pylori colonization is a definite risk factor for developing adenocarcinomas of the distal or non-cardia stomach, that's the lower part of the stomach, away from the junction with the esophagus. The World Health Organization classified H. pylori as a type I carcinogen back in 1994.
A definite carcinogen.
Yes. And again, in animal models, long-term experimental infection of gerbils with H. pylori can also result in gastric adenocarcinoma, reinforcing that causal link scientifically. As we discussed in detail, H. pylori can also induce primary gastric lymphoma, specifically the gastric malt lymphoma. While this condition is much less common than adenocarcinoma, it's clinically very important because of its unique response to treatment. Many of these diagnosed low-grade gastric B-cell lymphomas are, as we said, dependent on H. pylori for their continued growth. Crucially, these tumors may regress either fully or partially after H. pylori eradication. However, and this is important, they do require careful short and long-term monitoring after treatment. Any tumor that's not confined just to the superficial lining of the stomach, or even some that are but don't fully regress after eradication, may require additional treatment with chemotherapeutic agents or radiotherapy. It's not always just antibiotics and done. Next, let's consider functional dyspepsia. This is a very, very common condition where patients experience upper gastrointestinal symptoms, things like bothersome bloating, discomfort after eating, early fullness, sometimes upper abdominal pain. But when they undergo upper gastrointestinal endoscopy, the results are completely normal. No ulcer, no inflammation visible to the naked eye. It's sometimes called non-ulcer dyspepsia.
And H. pylori's role here.
Well, since H. pylori colonization is so common in the general population, a certain percentage of these patients with functional dyspepsia will naturally be colonized with the organism, just by chance. What's interesting here is that multiple clinical trials and meta-analyses have shown that H. pylori eradication therapy leads to symptom resolution in a small but statistically significant subgroup of these patients.
How small a subgroup?
We're talking maybe up to 15% more commonly than you see with a placebo treatment. It's still debated whether these responders might have had subtle peptic ulcers that were missed or in remission at the time of their endoscopy, or if there's a truly distinct small subgroup of functional dyspepsia patients whose symptoms are directly driven by H. pylori in some way we don't fully understand yet.
So should we treat it in functional dyspepsia?
Well, because functional dyspepsia can often be persistent and quite challenging to treat effectively with other means, most consensus conference guidelines actually recommend testing for and treating H. pylori in these patients if it's found. However, if you follow this advice, it's really important to realize and to communicate to the patient that only a relatively small subgroup of those treated will actually experience a benefit directly attributable to the eradication therapy itself. The number needed to treat to benefit one patient is often estimated to be around 14 or 15. So you treat 14 people to help one. It's something for both patients and doctors to keep firmly in mind when making that treatment decision.
So that covers the main bad news, the diseases we typically associate with H. pylori. But now for the really fascinating twist, the good news, the paradoxical protection that H. pylori seems to offer against certain other conditions. This is where it gets really interesting, almost flipping our usual understanding of this bug on its head.
It truly is a remarkable paradox and a significant area of recent research interest. It really forces us to rethink simple cause and effect relationships when it comes to microbes we've lived with for so long. A lot of attention has been focused on a potential protective role for H. pylori against complications of gastroesophageal reflux disease, or GERD, that's chronic heartburn. including Barrett's esophagus, which is a precancerous condition in the esophagus caused by chronic acid reflux, and critically, adenocarcinoma of the esophagus, and the gastricardia, which is a type of cancer right at the junction of the esophagus and stomach. This is quite counterintuitive to what many people might initially think a stomach bug protecting the esophagus.
Yeah, it seems backward. What's the evidence for this protection?
We have several compelling lines of evidence supporting this protective role. First, there's a striking temporal relationship. As the prevalence of gastric H. pylori colonization has steadily fallen in developed countries over the past several decades, we've simultaneously seen a corresponding dramatic rise in the incidence of these esophageal conditions, GERD, Barrett's, esophageal adenocarcinoma. It's almost as if one goes down, the other goes up, suggesting an inverse link.
An inverse correlation.
Exactly. Second, in most epidemiological studies, the prevalence of H. pylori colonization, particularly with those pro-inflammatory KAGE-positive strains, the very ones we associated with increased risk of gastric cancer, is significantly lower among patients who have these esophageal diseases compared to control participants without them. It's almost as if having H. pylori, especially the bad strains for the stomach, somehow protects your esophagus.
That's really odd.
It is. And third, prospective studies, following people over time, have shown an inverse relationship. The presence of H. pylori at the start of the study is inversely related to the subsequent development of these esophageal cancers. Having it seems to reduce the risk down the line.
So it's almost like a live by the sword, die by the sword situation, but in reverse. The same bacterium that might cause stomach issues could be preventing esophageal ones. How does that protective mechanism actually work? How does this little bug in the stomach act like a guardian for the esophagus?
The likely mechanism underlying this protective effect seems to be H. pylori-induced hypochlorhydria. Remember we talked about how in some patterns of gastritis, particularly pangastritis or corpus predominant gastritis, H. pylori colonization can lead to gastric atrophy and a significant reduction in stomach acid production over time.
Right, less stomach acid.
Exactly. This reduction in stomach acid or hypochlorhydria means there's simply less acidic refluxate reaching the lower esophagus. Less acid splashing up means less chronic irritation and damage to the esophageal lining over the years. And it's that chronic acid damage that's thought to drive the progression from GERD to Barrett's esophagus and ultimately to esophageal adenocarcinoma. So by reducing stomach acid, H. pylori indirectly protects the esophagus from acid injury. However, there's crucial clinical nuance here for practitioners managing individual patients. At the individual level, the severity of GERD symptoms can actually decrease, worsen, or remain completely unchanged after H. pylori treatment. It's not predictable for a single person. So, while the population-level data strongly suggests protection, concerns about potentially worsening GERD should not, as a general rule, override decisions about whether to treat H. pylori in an individual patient if a clear-cut indication for treatment exists, like they have a peptic ulcer or multiple lymphoma. You treat the definite problem.
Makes sense.
If, however, there is no clear indication for H. pylori treatment, then clinicians must carefully balance the potential benefits of eradication, like maybe reducing long-term gastric cancer risk, though that's debated in asymptomatic people, against the potential harms, including the small but real risk of unmasking or worsening GERD symptoms after treatment. It becomes a much more complex risk-benefit assessment for each person in that situation.
It's a crucial point for practitioners. Don't let the potential protective effects outweigh clear indications for treatment, but certainly be aware of them when the indication is less clear. Are there any other surprising protective effects or other body-wide associations linked to H. pylori, maybe beyond just the gut and esophagus?
Yes. H. pylori has an increasingly recognized role or association with other gastric pathologies and even some conditions outside the digestive tract entirely. For instance, it may predispose some patients to iron deficiency, leading to iron deficiency anemia.
How does it cause iron deficiency?
It's thought to happen through a couple of mechanisms. One is potential occult or hidden blood loss from the chronic low-level inflammation in the stomach lining over many years. The other, perhaps more common mechanism, is through H. pylori-induced hypochlorhydria that reduce stomach acid again. Stomach acid is actually important for converting dietary iron into a form that can be absorbed efficiently in the small intestine. So if your stomach acid is low due to H. pylori, you absorb less iron from your food, potentially leading to deficiency over time. Looking beyond the GI tract, there's been quite consistent reporting regarding idiopathic thrombocytopenic purpura, ITP. This is an autoimmune condition where the body mistakenly attacks its own platelets, which are crucial for blood clotting, leading to abnormally low platelet counts and a risk of bleeding. Numerous studies, really quite consistently now, have described significant improvement in, or even complete normalization of, platelet counts after successful H. pylori eradication in a subset of patients with ITP. The mechanism isn't fully understood, maybe some molecular mimicry triggering the autoimmunity, but the clinical observation is quite strong, and eradication is often tried in ITP patients found to have H. pylori.
Wow, that's remarkable. An autoimmune condition improving after antibiotic treatment for a stomach bug.
It is quite remarkable. Now, there are other potentially important but even more controversial associations, and it's really important to approach these with a healthy dose of caution. These include a potential protective link with ischemic heart disease and cerebrovascular disease, basically heart attacks and strokes. Some early studies suggested an inverse association.
Protection against heart disease, that seems unlikely.
Well, the strength of these associations significantly diminishes or disappears altogether when confounding factors, other lifestyle or health factors that travel with H. pylori status, are carefully taken into account in the analysis. Our current knowledge here is incomplete, and frankly, most authorities generally consider these associations to be non-causal. It's unlikely H. pylori is directly protecting the heart or brain in a meaningful way. Perhaps most intriguing, though, and a topic that really ties back to that concept of our long coevolution with this bacterium, is the growing body of studies showing an inverse association of Kaga-positive H. pylori with childhood-onset asthma, hay fever, and other atopic disorders, basically, allergies and allergic diseases.
So having H. pylori might protect against allergies.
That's what the evidence increasingly suggests. Multiple epidemiological studies have found that children colonized with H. pylori, especially the Kage-positive strains, are less likely to develop these allergic conditions compared to children without H. pylori.
Why would that be?
The leading hypothesis relates to the hygiene hypothesis, or old friends hypothesis. The idea is that early life exposure to certain microbes, like H. pylori, helps to properly train the developing immune system, steering it away from the overreactive allergic type responses that characterize asthma and allergies. H. pylori might modulate the immune system in a way that promotes tolerance. And importantly, these associations have even been shown to be causal in relevant animal models. Studies in mice, for example, have demonstrated that inducing H. pylori colonization early in life can actually prevent the development of experimentally induced allergic airway inflammation, mimicking asthma.
So there's a biological mechanism.
There seems to be, yes, demonstrated in animal models. While the exact effect size and mechanisms in humans are still being actively investigated and haven't been definitively established yet, it paints a really fascinating picture. It suggests a world where our very success in reducing exposure to and eradicating a long-standing microbial partner like H. pylori might be inadvertently leaving us more vulnerable to a whole new set of modern health challenges, like the epidemic of allergic diseases we're seeing. It really makes you ponder how the disappearance of this ancient co-colonizer might be subtly reshaping the landscape of modern immune-related diseases.
That truly frames H. pylori as this incredibly complex biological entity, doesn't it? Capable of both significant harm and, quite surprisingly, potential protection. So, given this intricate nature, if clinicians suspect H. pylori is involved in a patient's condition, how do they actually diagnose it? What are the standard approaches they use to find out if it's there?
Right. Diagnosing H. pylori accurately is obviously key. Broadly speaking, the available tests fall into two main groups. There are those who require the patient to undergo an upper gastrointestinal endoscopy, where a small flexible camera is inserted through the mouth to look directly at the stomach lining.
The invasive ones.
Exactly. And then there are simpler, non-invasive tests that can be performed more readily in a clinic or lab setting without needing that procedure.
Yeah.
Let's start with the endoscopy-based tests. Now, endoscopy isn't generally considered necessary for the initial management of young patients, say under 45 or 50, depending on local guidelines, who present with simple indigestion or dyspepsia without any worrying features.
Okay.
However, it becomes commonly used, and indeed quite important, for older patients or for any patient who presents with what we call alarm symptoms.
Alarm symptoms, like what?
These are red flags that might suggest something more serious is going on, like cancer. Things like unexplained weight loss, difficulty swallowing, dysphagia, persistent vomiting, signs of bleeding like vomiting blood or black stools, or a new onset of iron deficiency anemia. In these situations, endoscopy is crucial primarily to rule out serious conditions like gastric cancer, but it also provides an opportunity to make a positive diagnosis of H. pylori if it's present by taking biopsies. If an endoscopy is performed, several tests can be done on the biopsy samples. The most convenient and widely used biopsy-based test is the biopsy-urease test, sometimes called the C-LOTUS.
How does that work?
Here, one small piece of gastric tissue, or maybe two smaller ones, are taken from the stomach lining during the endoscopy and immediately placed into a small well containing a gel. This gel contains urea and a pH indicator chemical. If H. pylori urease, that special enzyme we talked about, is present in the biopsy tissue, it will rapidly break down the urea in the gel, producing ammonia. This ammonia raises the pH, which causes a distinct color change in the indicator, usually from yellow to pink or red.
It's quick.
It can be very quick, often occurring within minutes if the bacterial load is high, though it's usually read up to 24 hours, just in case. It's a very practical test in the endoscopy suite. It's worth noting that those rarer non-pylori helicobacters, if present, may also give weakly positive results in this test, though usually much slower. Another highly accurate method using biopsies is histologic examination. This involves sending the biopsy specimens to the pathology lab, where they're processed and stained so a pathologist can look for the bacteria under a microscope.
What do they look for?
For optimal visualization of the organism, a special stain is usually required, such as a modified Giemsa stain, a silver stain, or an immunostain using antibodies against H. pylori. These help the characteristic curved or spiral-shaped bacteria stand out against the background tissue. A significant advantage of histologic study, particularly if biopsy specimens are obtained from both the antrum, lower stomach, and the corpus, upper stomach, is that it yields additional, absolutely crucial information beyond just presence-absence of the bug. The pathologist can assess the degree and pattern of inflammation, gastritis, and critically look for the presence of any atrophy, thinning of the lining, intestinal metaplasia, cell type changes, or dysplasia, precancerous changes. This information is vital for assessing future cancer risk.
So you get much more than just a yes/no for H. pylori.
Exactly. And if those non-pylori gastric helicobacters happen to be present instead, they can sometimes be visualized by the pathologist as characteristic long, tightly coiled spirals, which look different from H. pylori. Finally, within the endoscopy-based group, there's microbiologic culture. This involves taking biopsy specimens and trying to grow the H. pylori in the lab on special culture media.
Growing the actual bacteria.
Yes. While culture is theoretically the most specific test, if you grow it, it's H. pylori, it can sometimes be insensitive in practice. H. pylori is a notoriously difficult organism to isolate and grow in culture. It's quite fastidious. However, if the organism is successfully cultured, its identity as H. pylori can be confirmed by its typical appearance under a microscope after gram stain, its gram-negative curved, and its positive reactions in standard biochemical tests like oxidase, catalase, and urease. But the crucial advantage of culture, when successful, is that it allows for the determination of the organism's antibiotic susceptibility.
Testing which antibiotics work against it.
Precisely. The lab can test the cultured bacteria against a panel of relevant antibiotics, like clarithromycin, metronidazole, levofloxacin, to see which ones it's resistant or sensitive to. This information can be incredibly useful clinically, especially in managing difficult cases where initial eradication treatments have failed. It allows for targeted therapy.
So endoscopy offers a lot of detailed information, especially with histology and potentially culture. But for many patients, particularly younger ones without alarm symptoms, we're probably not jumping straight to an endoscopy, right?
Yeah.
What are the non-invasive options that are more commonly used for initial diagnosis or screening?
You're absolutely right. For routine screening or diagnosis, when gastric cancer doesn't need to be explicitly ruled out by direct visualization with endoscopy, non-invasive H. pylori testing is the norm. These tests are much more convenient and less risky for the patient. The longest established and a very accurate non-invasive test is the urea breath test, often abbreviated as a UBT.
How does the breath test work?
It's quite elegant, actually. The patient drinks a small amount of the solution or swallows a capsule containing urea that's been labeled with a non-radioactive stable isotope of carbon, usually carbon-13, 13C, or sometimes carbon-14, 14C, though 13C is preferred as it's non-radioactive. Then, after a short wait, they simply blow into a collection tube or bag. If H. pylori urease is present in their stomach, it will break down the labeled urea, just like in the biopsy urease test. This release is labeled carbon dioxide, 13CO2 or 14CO2. This labeled CO2 is absorbed into the bloodstream, travels to the lungs, and is exhaled in the breath, where it can be detected by a special analyzer. If no H. pylori urease is present, the labeled urea passes through undigested, and no labeled CO2 appears in the breath.
Clever. Any special requirements?
Yes, this test does require the patient to fast for several hours beforehand, usually overnight, to ensure accuracy, as food in the stomach can interfere. It's highly accurate, though, often considered the gold standard non-invasive test by many. Another very popular non-invasive option is the stool antigen test. This is exactly what it sounds like. It detects H. pylori antigens, essentially fragments or proteins of the bacterium in the patient's stool sample.
How accurate is that?
Modern stool antigen tests, particularly those that use monoclonal antibodies, which are highly specific, are also very accurate, nearly as accurate as the urea breath test. They are generally considered simpler to perform logistically, more convenient for many settings, and often less expensive than the breath test. The main drawback, perhaps, is patient acceptance, as you can imagine. Some patients might find collecting a stool sample undesirable or inconvenient compared to blowing in a tube.
Fair point. What about blood tests? Are they useful?
Yes. The third category of non-invasive tests is serologic assays, which are blood tests. These are perhaps the simplest tests from a patient perspective, just requiring a blood draw. They measure specific IgG antibody levels in the patient's blood serum. The body produces these antibodies in response to the H. pylori infection.
Are they accurate?
Here's where it gets a bit tricky. The best laboratory-based serologic tests, typically using validated enzyme-linked immunosorbent assay, ELISA, or immunoblot methods, can be nearly as accurate as the breath or stool tests for detecting current or past infection. However, a significant caveat here is that many commercially available serologic tests, especially the rapid point-of-care or office-based tests, do not perform well or provide consistently accurate results. Their sensitivity and specificity can be quite variable. So, selecting a reliable serologic test, usually one performed by a reputable clinical laboratory, is absolutely key if you're going this route.
Okay, reliability varies. Any other pros or cons of blood tests?
One potential advantage of serology is that antibody levels are generally less affected by recent antibiotic use or by proton pump inhibitors, PPIs, compared to the breath and stool tests. Breath and stool tests rely on the presence of active, metabolically functioning bacteria producing urease or shedding antigens. Recent antibiotics, or PPIs, can suppress the bacteria temporarily, leading to false negative results on breath or stool tests. Serology, measuring the host's immune memory, is less susceptible to this short-term suppression. However, the flip side is that antibodies can persist for months or even years after the infection has been successfully eradicated.
So a positive blood test doesn't necessarily mean active infection.
Exactly. It tells you the person has been infected at some point and likely still is if they haven't been treated, but it's not ideal for confirming eradication after treatment, as we'll discuss.
That's a comprehensive look at how we initially diagnose H. pylori. But let's say someone is treated for H. pylori. How do clinicians then confirm that the treatment was actually successful in getting rid of the bug? Do we just rerun the same tests? Seems like that persistence of antibodies could be a problem for blood tests.
That's a crucial clinical question because confirming eradication is vital, especially for patients treated for conditions like peptic ulcers or multiple lymphoma, where relapse is a concern if the bug isn't gone. Yes, the urea breath test, the stool antigen test, and even biopsy-based tests, if endoscopy is being repeated for another reason like checking ulcer healing, can all be used to assess the success of eradication treatment. However, there's a very important caveat that clinicians must be acutely aware of, a critical point for practice, because the breath test, stool test, and biopsy urease test are all dependent on the H. pylori bacterial load, the number of active bacteria present.
Right.
Using them too soon after completing the antibiotic treatment course can yield false negative results.
Too soon? How soon is too soon?
Generally, testing should be delayed until at least 4 weeks after the antibiotic therapy is finished. The reason is that the initial antibiotic treatment might just suppress the bacterial numbers temporarily, reducing them below the level of detection for these tests, but not actually eliminate them completely. If you test too early, you might get a negative result, think you're cured, but then the organism might regrow weeks or months later. It's like sweeping dirt under the rug. It looks clean, but the problem is still there. For the same reason, these load-dependent tests, breath, stool, biopsy, urease, are unreliable if they are performed within 4 weeks of any intercurrent treatment with other antibiotics, taken for unrelated reasons, or bismuth compounds like Pepto-Bismol. Also, and this is very important in practice, they are unreliable if performed within 1 to 2 weeks of the patient discontinuing proton pump inhibitor, PPI treatment.
PPIs interfere too.
Yes, PPIs, by significantly reducing stomach acid, create a less favorable environment for H. pylori and can also suppress the bacterial numbers enough to cause a false negative on these tests, even without antibiotics. So, patients usually need to stop their PPI for at least 1, ideally 2, weeks before having a breath test or a stool test to check for eradication.
It's a lot of timing
It
considerations.
is, and it's critical for accurate results.
Yeah.
In most situations, non-invasive tests, the urea breath test or the stool antigen test, are preferred for assessing treatment success due to their convenience and accuracy when timed correctly. However, there's a specific scenario. After treatment for gastric ulceration, not usually duodenal, it's generally recommended that endoscopy be repeated anyway, maybe 6 to 8 weeks later. This is done not just to assess H. pylori eradication, but primarily to ensure the ulcer itself is fully healed, and critically to rebiopsy the ulcer site or scar to exclude any underlying gastric carcinoma, which is a higher risk with gastric ulcers than duodenal ones. If PPIs have been stopped for at least 2 weeks before this repeat endoscopy, and no antibiotics or bismuth compounds have been given for at least 4, preferably 6, weeks, then this endoscopy offers a valuable opportunity to assess treatment success directly using biopsy-based tests, urease or histology, alongside checking ulcer healing.
And what about the blood tests for checking success? You hinted they weren't ideal.
You're right. Serologic tests, the ones that measure antibody levels in the blood, are not generally used to monitor treatment success or confirm eradication. The reason is simple. Antibody levels decline very slowly after the infection is successfully cleared. It can take many months, sometimes even a year or longer, for IgG antibody levels to fall significantly enough to indicate successful treatment reliably. This makes them impractical for timely confirmation of cure shortly after therapy. You can't use them for a quick check a month or 2 later.
Okay, that makes sense. So we have identified the bug, confirmed its presence, and now we know how to check if our treatment worked. With careful timing, that brings us squarely to the core of clinical management, the treatment strategies themselves. When is treatment for H. pylori actually indicated? Is it for everyone who tests positive or only in specific scenarios? This seems key, given the potential downsides and protective effects we discussed.
That's a vital question, and the answer is definitely not everyone who tests positive needs treatment. Understanding the clear indications is paramount, especially given the potential for antibiotic side effects, resistance, and altering the microbiome. The most clear-cut, universally accepted indications for eradication treatment are for patients with documented H. pylori-related duodenal or gastric ulceration. This applies whether the ulcers are currently active or if there's just a history of documented peptic ulcer disease. Eradicating H. pylori in these cases is proven to dramatically reduce the risk of ulcer recurrence, often providing a permanent cure for their ulcer disease. That's a huge benefit. The other clear-cut indication is for patients diagnosed with low-grade gastric B-cell lymphoma, that rare type of stomach lymphoma we discussed that arises due to chronic H. pylori stimulation and often responds dramatically to eradication therapy. Treatment is standard of care here.
Okay, ulcers and molt lymphoma, definite treatment. What about less clear-cut situations?
Beyond these definite indications, guidelines have also generally recommended H. pylori treatment for colonized patients who meet the criteria for functional dyspepsia, those upper GI symptoms with a normal endoscopy.
Even though only a small percentage benefit.
Yes, the recommendation comes with that significant caveat. It's made largely on the basis that some patients, that small subgroup, maybe 1 in 14, will benefit symptomatically, and functional dyspepsia can be difficult to manage otherwise. But it's not a guaranteed fix for their symptoms by any means. Then there are conditions for which H. pylori eradication has been recommended by some guidelines or experts, but it's perhaps not as universally supported as the definitive causal link to H. pylori, or the consistency of response to eradication isn't quite as robust as for ulcers. These include idiopathic thrombocytopenic purpura, ITP, where platelet counts may improve, unexplained vitamin B12 deficiency, and unexplained iron deficiency anemia. For these last two, treatment might be considered, but only after other potential causes for the deficiencies have been carefully and thoroughly excluded. You wouldn't jump to treating H. pylori for anemia if the person clearly has another reason for it.
Right, rule out other causes first. What about preventing cancer in high-risk people?
That's another area.
Yeah.
For individuals with a strong family history of gastric cancer, or perhaps those found to have extensive gastric atrophy or intestinal metaplasia on biopsy, known precursor conditions, treating H. pylori in the hope of reducing their future cancer risk is considered reasonable by many experts and some guidelines.
But is the benefit proven?
It's still somewhat unproven in terms of hard outcomes like mortality. While several meta-analyses of randomized trials do show that H. pylori eradication modestly reduces the future incidence of gastric cancer compared to no treatment, especially if treated before advanced precancerous changes develop, There's no current definitive evidence from these trials to suggest it reduces all-cause mortality in these asymptomatic individuals. So it's a nuanced decision, often based on assessing individual risk factors and patient preferences, rather than a universally mandated prevention strategy with guaranteed life-saving benefit.
That lays out the why of treatment very clearly. Now let's talk about the who and how of diagnosis and treatment in the real world of clinical practice. How do clinicians typically decide on the management flow for different types of patients presenting with symptoms, like indigestion?
This is where clinical algorithms and guidelines come into play, especially for patients presenting with indigestion or dyspepsia, which is incredibly common. The approach often differs based on age and the presence of those alarm symptoms we mentioned. For older dyspeptic patients, the age cutoff varies, but often 50 or 60, depending on local cancer risk. Or for any patient who presents with any of those alarm symptoms, weight loss, bleeding, swallowing difficulty, etc., an upper gastrointestinal endoscopy is generally indicated as the first step.
Why endoscopy first?
The primary purpose here is twofold. First, to seek a definitive diagnosis for their symptoms, and critically, to rule out malignancy like gastric or esophageal cancer, which becomes more common with age and alarm features. Second, during that endoscopy, biopsies can be taken to test for H. pylori. The decision to eradicate the organism, if found, is then based on whether a clear indication, like an ulcer or severe gastritis, is discovered during that procedure. Now, for young dyspeptic patients in the community who have no alarm symptoms, endoscopy is generally considered unnecessary as an initial step. It's invasive and expensive, and the risk of serious underlying pathology like cancer is very low in this group.
So what's the approach for them?
It often depends on the background prevalence of H. pylori in that particular community. If the community prevalence is known to be low, say below approximately 15–20%, the initial approach recommended by many guidelines for these young non-alarm patients is often just a short empirical course of acid suppression using a proton pump inhibitor, PPI, to see if symptoms resolve.
And if that doesn't work, or if prevalence is high?
Right. If these patients don't respond adequately to the PPI treatment, or if their symptoms relapse soon after stopping the PPI, or if the community prevalence of H. pylori is known to be higher, above 20%, then most guidelines recommend a strategy of non-invasively testing for H. pylori, using breath or stool tests, and then eradicating it if it's found. This is often called a test and treat strategy.
What's the rationale for test and treat?
The rationale is primarily to identify and cure the patients within that group who do have underlying undiagnosed peptic ulcer disease causing their symptoms. It also aims to benefit that small percentage, maybe 5% to 10% of patients whose functional dyspepsia symptoms will genuinely respond to H. pylori eradication. However, it's important to acknowledge again that the majority of patients treated under this strategy solely for functional dyspepsia will likely not experience a direct benefit from the eradication itself, beyond any placebo effect.
So, while test and treat is a guideline strategy in some situations, this isn't a call for widespread population screening of everyone, is it? We're not advocating testing every single person for H. pylori just in case they have it.
Precisely. That's a really important distinction. Currently, widespread community screening for H. pylori and treatment of all positives, often called screen and treat, as a primary public health strategy for preventing gastric cancer and peptic ulcers is not recommended in most countries.
Why not, if it causes cancer and ulcers?
There are several compelling reasons for this cautious stance. First, as we discussed, the full extent of the consequent reduction in gastric cancer risk following eradication in asymptomatic individuals is not yet definitively known or quantified, especially regarding impact on overall mortality. While studies show a modest reduction in cancer incidence, the follow-up periods are still relatively short in the grand scheme of cancer development, and the magnitude of the effect might vary significantly in different populations with different baseline risks. It's not a magic bullet for cancer prevention yet. Second, there are definite downsides to mass treatment. These include the common and sometimes severe rare adverse side effects associated with the multi-drug antibiotic regimens used, like diarrhea, taste disturbance, allergic reactions. Third, there's the major public health concern regarding the emergence of antibiotic resistance, which may develop not only in H. pylori itself, making future treatments harder, but also potentially in other incidentally carried bacteria within the body, contributing to the broader societal problem of antibiotic resistance. Other reasons. Yes. Fourth, there's the potential for inducing anxiety or disease labeling in otherwise healthy people if they are told they have a chronic bacterial infection, especially if the treatment is complex or unsuccessful. And finally, as we've discussed, There's the existence of that subset of people who will actually develop new GERD symptoms or see their existing GERD worsen after H. pylori treatment due to the loss of its acid-suppressing effect in some individuals. So, when you weigh the uncertain long-term benefits against these definite potential harms and costs, the balance currently tips against mass screening and treatment for asymptomatic individuals in most settings.
Despite the strong arguments against widespread screening and treatment of asymptomatic people, it seems like, anecdotally at least, some doctors still do treat H. pylori if it's found incidentally, even in asymptomatic carriers, especially maybe in younger individuals or children. What's the rationale driving that, and is it really evidence-based practice?
That's a very pertinent observation about real-world practice versus guidelines. It's true, it's a common clinical dilemma. Despite the general absence of formal screening strategies or guideline recommendations for treating asymptomatic carriage, it's undeniable that many doctors do choose to treat H. pylori if it's known to be present, perhaps found during investigation for something else, particularly in children and younger adults, even when the patient has absolutely no symptoms related to it.
Why do they do it then?
The rationale often stems from a few places. Sometimes it's simply to reduce patient concern or anxiety about harboring a known carcinogen. Other times, it's driven by the hope for a potential future reduction in gastric cancer risk, with the thinking that any such reduction is likely to be greater if the infection is eradicated earlier in life, before decades of damage have accumulated. The idea is, get it while they're young.
Does that sound reasoning?
Well, here's the critical counterpoint. Such practices, almost universally, do not factor in any potential benefits of lifelong H. pylori colonization that we've just discussed, like the potential protection against esophageal adenocarcinoma or possibly against asthma and allergies. They focus solely on the potential negatives of H. pylori. So overall, despite this being a widespread clinical activity in some areas, it's crucial to emphasize that most treatment of persons with asymptomatic H. pylori carriage is currently given with no firm evidence base definitively demonstrating a net benefit, balancing pros and cons, for that individual patient's long-term health or mortality. It really highlights a gap sometimes between current scientific knowledge, guideline recommendations, and clinical practice, which can sometimes be driven more by a better safe than sorry approach or patient request than by solid data in this specific asymptomatic context.
Okay, let's return to a specific firm indication where treatment is strongly recommended. Patients diagnosed with low-grade B-cell lymphomas, what's the approach there?
Right, the cancer that responds to antibiotics? Exactly. For these patients, H. pylori eradication therapy should be attempted in essentially all cases, regardless of whether H. pylori can actually be detected by the diagnostic tests used at the time of diagnosis.
Even if the tests are negative?
Yes. This is because there can be false negative results for H. pylori testing, especially if the patient has recently been on PPIs or antibiotics, or if the bacterial load is low in the context of the lymphoma. And given that up to 70%, maybe even more, of these patients experience significant tumor regression after eradication, it's considered worth trying the treatment, even if the bug isn't immediately obvious. The potential benefit is huge. It's important to remember, as we discussed, that not all of these cases diagnosed morphologically as low-grade malt lymphoma necessarily represent true irreversible malignancies. The reported high success rate of eradication therapy may sometimes reflect the cure of what were actually benign, but potentially pre-malignant lymphoid processes that just look like a tumor under the microscope. For cases suspected of being true malignancies, examination of the tumor tissue for characteristic chromosomal translocations, like that T1118, should ideally be done. This can help distinguish truly malignant processes from reactive ones, and can help guide decisions about further therapeutic approaches if eradication alone isn't sufficient. Fortunately, these low-grade malt tumors are generally very slowly progressive, which usually allows sufficient time for H. pylori eradication therapy to be given and its effect evaluated over several months, without unduly delaying the potential need for later chemotherapy or radiotherapy if the tumor persists or progresses.
That's a deeply nuanced understanding of when and why to treat. Now let's finally get down to the brass tacks, the specific treatment regimens themselves. If a decision is made to treat, what does an effective H. pylori eradication therapy actually look like? Are we talking about just prescribing a single standard antibiotic?
No, absolutely not. Just a single antibiotic. That's a really important point. While H. pylori is actually susceptible to a wide range of different antibiotics in vitro, meaning if you test it in the lab dish monotherapy, or using just one antibiotic alone is almost never successful in vivo in a living person. It consistently fails.
Why does monotherapy fail if the bug is susceptible in the lab?
It's likely due to a combination of factors, including the difficulty of achieving high enough concentrations of a single active antibiotic within that specific colonization niche deep in the gastric mucus, possibly even inside cells, which is a tough place for drugs to penetrate effectively and stay active. Also, resistance can emerge very quickly if only one drug is used. Therefore, current standard H. pylori eradication regimens are always multi-drug regimens. Typically, they consist of a proton pump inhibitor, PPI, that's a drug like omeprazole, lansoprazole, etc., primarily for strong acid suppression, along with 2 or sometimes 3 different antimicrobial agents, antibiotics or bismuth. And these combinations are generally given for a duration of 10 to 14 days. Longer courses tend to be slightly more effective than shorter ones, like 7 days. Now, it's absolutely vital to understand that the optimal regimens actually vary significantly in different parts of the world. This variation is primarily driven by the known local or regional rates of primary antibiotic resistance in the circulating H. pylori strains. Resistance patterns are not uniform globally.
So what works well in one country might not work well elsewhere.
Precisely. Because of this, guidelines on the optimal first-line and second-line regimens for H. pylori eradication are constantly evolving in individual countries or regions based on ongoing surveillance of resistance patterns. Therefore, clinicians must always refer to the most up-to-date local guidelines for their specific geographic area when choosing a regimen. This is crucial for maximizing the chances of successful treatment on the first attempt. Using an outdated or inappropriate regimen for the local resistance situation is a common reason for treatment failure.
So regional resistance patterns are key. Beyond choosing the right regimen for the region, what are the 2 most important factors that determine whether that chosen treatment regimen will actually be successful for an individual patient? What does it truly boil down to for success at the patient level?
You've hit on the 2 most critical points for success, assuming an appropriate regimen was chosen initially. The first and arguably most important factor in achieving successful H. pylori eradication is the patient's close compliance with the prescribed regimen.
Taking all the pills.
Exactly. Taking all the medications exactly as prescribed, usually multiple pills, multiple times a day, for the full 10 or 14-day duration, even if they start feeling better partway through. Treatment failure following even relatively minor lapses in compliance is unfortunately common. And perhaps even more concerning, such lapses often lead to the acquisition of secondary antibiotic resistance by the surviving H. pylori strain, making future treatment attempts much, much harder.
So compliance is paramount. How can we ensure it?
Well, to stress the importance of compliance, it's essential that clear, preferably written, instructions be given to the patient. And it's also crucial that common minor side effects of the regimen, which are often expected, like maybe an metallic taste, some nausea, or mild diarrhea, are thoroughly explained beforehand. If patients know what to expect, they're less likely to stop the treatment prematurely if they experience these manageable side effects. Good communication is key. The second most important factor for success is, as we've touched on, the use of drugs to which the patient's particular H. pylori strain has not already acquired resistance. This brings us back squarely to the growing global concern regarding antibiotic resistance.
Which antibiotics are most affected by resistance?
Increasing levels of primary H. pylori resistance, meaning resistance in people who've never been treated for H. pylori before, to certain classes of antibiotics are of particular concern worldwide. The main culprits are macrolides, specifically clarithromycin, and fluoroquinolones, like levofloxacin. Resistance to metronidazole is also quite common in many areas, though perhaps slightly less impactful on treatment success than clarithromycin resistance. In most parts of the world now, with the main exception perhaps being northwestern Europe, where resistance rates have historically been lower, the rate of primary clarithromycin resistance is sufficiently high, often exceeding 15 to 20%, that regimens containing clarithromycin plus only one other antibiotic, like the classic PPI plus clarithromycin plus amoxicillin triple therapy, often fail to achieve acceptable eradication rates, usually 85 to 90%. They just don't work well enough anymore in many places as first-line therapy.
So clarithromycin triple therapy is often out. What's used instead?
Well, regimens that include clarithromycin but combine it with 2 other antibiotics, like concomitant therapy, which is PPI plus clarithromycin plus amoxicillin plus metronidazole altogether, may still remain an option, as the other 2 antibiotics are more likely to eradicate the H. pylori, even if the strain happens to have developed clarithromycin resistance. Or, more commonly now, in areas with high clarithromycin resistance, the preferred first-line therapy might be a bismuth quadruple therapy, PPI plus bismuth plus tetracycline plus metronidazole. A general rule of thumb that clinicians should follow regarding resistance is that when a patient is known to have had even remote prior exposure to clarithromycin or a fluoroquinolone like levofloxacin, perhaps for a respiratory infection years ago, these specific antibiotics should generally be avoided in subsequent H. pylori eradication regimens for that patient, as the risk of resistance is much higher.
What about other common antibiotics like amoxicillin or tetracycline?
Fortunately, significant resistance to amoxicillin or tetracycline remains unusual in most parts of the world, even if these antibiotics have been given previously for other infections. Similarly, resistance to metronidazole, when it occurs, is often only partial and can sometimes be overcome by using higher doses or longer duration. Therefore, there's typically no need to avoid using amoxicillin, tetracycline, or metronidazole in H. pylori regimens, regardless of whether they have been previously prescribed to the patient for other reasons. They are generally more reliable backbones. One other point about optimizing regimens. Regardless of which specific antibiotic combination is chosen, meta-analyses of clinical trials have consistently shown that using high rather than standard doses of acid-suppressive PPIs in conjunction with the antibiotics significantly increases the effectiveness of the eradication regimen. Getting profound acid suppression seems to help the antibiotics work better, perhaps by making the bacteria more vulnerable or improving antibiotic stability.
So higher PPI doses are better.
Generally, yes. Doubling the standard PPI dose is often recommended in eradication regimens. And looking to the future, there are newer, even more potent acid suppressants being developed. For instance, vonoprazan, which is a potassium-competitive acid blocker, PCAB, is licensed in Japan and some other countries. Studies have shown it's associated with higher H. pylori eradication rates when used in conjunction with amoxicillin and clarithromycin compared to when a standard PPI was used for acid suppression. PCABs might represent a future shift in the acid suppression component of the gepfromations as they become more widely available.
So given the rising resistance issues and the need for precision, is antibiotic susceptibility testing actually testing the patient's specific H. pylori strain for resistance before starting treatment, something we should be doing routinely? It sounds like it would be the ideal approach, almost like a pre-flight check for the bacteria.
In an ideal world, assessing antibiotic susceptibilities before initiating first-line treatment would absolutely be the optimal strategy. It would allow for truly personalized and targeted therapy, maximizing the chance of success on the first try and minimizing the use of ineffective antibiotics, which contributes to further resistance.
But it's not routinely done. Why not?
Correct. It is not usually undertaken in routine clinical practice for a few key practical reasons. First, as we discussed, obtaining H. pylori for culture and susceptibility testing currently requires the patient to undergo an endoscopy and mucosal biopsy, which is invasive, carries some risks, and is costly. Second, most routine clinical microbiology laboratories are relatively inexperienced in H. pylori culture due to its specific and rather challenging growth requirements. It's not as simple as culturing many other common bacteria, so the process can be difficult, specialized, and not widely available. So because of these hurdles, the standard approach, if initial empirically chosen H. pylori treatment fails, is usually empirical retreatment with another different standard drug regimen. A second-line regimen, often bismuth quadruple therapy if triple therapy failed or vice versa, or maybe a levofloxacin-containing regimen. This means choosing a new regimen based on general knowledge of common resistance patterns and what the patient received first, but still without specific susceptibility data for that patient's strain.
And if the second attempt fails too?
If that second-line empirical treatment also fails, then the third-line approach ideally should involve referral for endoscopy, biopsy, and culture with susceptibility testing, so that subsequent treatment can finally be based on documented antibiotic sensitivities for that patient's now proven difficult-to-treat strain. However, even at this stage, due to the practical challenges or lack of availability of reliable susceptibility testing, empirical third-line therapies, using drugs less commonly associated with resistance, like rifabutin perhaps, are still often used in practice. It remains a complex dance between ideal, personalized medicine and real-world constraints. Just to give listeners a flavor, though, common regimens recommended in guidelines, again varying by region and line of therapy, often fall into these categories. There's triple therapy, usually a PPI, plus clarithromycin, plus either amoxicillin, OCA, or metronidazole, OCM, typically for 14 days. Still viable first line only in areas with very low 15-20% clarithromycin resistance. There's bismuth quadruple therapy, OBTM, a PPI plus bismuth subsalicylate or subcitrate, plus tetracycline plus metronidazole, usually for 10-14 days.
Often used first line where clarithromycin resistance is high, or commonly as second line therapy. There's concomitant therapy, PPI, plus amoxicillin, plus clarithromycin, plus a metronidazole or tinidazole, all given together for 10-14 days. It's essentially giving all the drugs from the two main triple therapies at once. An alternative first-line to bismuth quad, and levofloxacin-containing regimens like PPI plus amoxicillin plus levofloxacin OAL, usually for 10-14 days. Mostly reserved for second or third line therapy due to rising fluoroquinolone resistance and potential side effects. Less effective when fluoroquinolone use is high, or if the patient had prior exposure. As mentioned for the PPI component in any of these, using higher doses, example, omeprazole 20 mg twice daily doubled to 40 mg twice daily, or using naturally more potent PPIs like isomeprazole or haprazole, is often recommended to maximize efficacy, especially for patients known to be extensive metabolizers of PPIs via the CYP2C19 enzyme pathway.
That's incredibly detailed and provides a real sense of the complexity and the careful decision-making process involved in treatment. It's clear that treating H. pylori effectively isn't just a simple prescription anymore. It requires real expertise and attention to local factors and patient history. So with all this accumulated knowledge, what does the future hold for H. pylori? What are the broader public health implications globally, and how might we think about preventing colonization on a larger scale in the future?
The public health significance of H. pylori remains profound, absolutely. In economically richer countries, it continues to be strongly associated with significant morbidity from peptic ulcer disease and mortality from gastric adenocarcinoma, even with falling prevalence. But its impact can be even more devastating in some economically poorer countries, where gastric adenocarcinoma, driven largely by H. pylori, may be an even more common cause of cancer death later in life compared to richer nations. So, it's truly a global health burden, though its specific manifestations and impact vary geographically. If we were to contemplate mass prevention strategies for the future, moving beyond just treating established infections, the most obvious and potentially impactful method would be vaccination.
A vaccine against H. pylori, is that feasible?
It's certainly been a major goal of research for decades. There's been promising research in this area. Experimental immunization of animals, including non-human primates, has yielded some encouraging results, showing protection against challenge infection. And the first reported large-scale clinical trial of an H. pylori vaccine in humans, conducted in children in China a few years ago, did show some efficacy, although perhaps not as high as hoped, in preventing acquisition. Further trials with improved vaccine candidates are reportedly ongoing or planned. A successful, effective, and safe vaccine could truly revolutionize global health by preventing colonization in the first place, potentially eradicating H. pylori-related diseases, much like we've done with other infectious diseases through vaccination. It remains a key goal.
A vaccine would undoubtedly be a game changer, potentially preventing a lot of suffering from ulcers and stomach cancer worldwide. But you've also spent a good amount of time highlighting H. pylori's unexpected protective roles, this double-edged sword aspect. This begs a really provocative question as we think about prevention. What are the potential biological and clinical costs of preventing this colonization entirely, especially for an organism that has literally co-evolved with humans for millennia? If we were successful in removing it completely from the human population, what else might we be inadvertently changing in ourselves or our susceptibility to other diseases?
That's precisely where the deeper implications lie, and it forces us to consider potential unintended consequences very carefully. It really challenges our traditional view of microbes solely as pathogens to be eradicated. It forces us to reconsider our complex, long-standing relationship with our ancient microbial partners. As we've discussed quite extensively, lifelong absence of H. pylori is now recognized as a definite risk factor for GERD complications, including Barrett's esophagus and the increasingly common esophageal adenocarcinoma, particularly adenocarcinoma of the gastrocardia. So if we were to hypothetically eradicate H. pylori from everyone globally through vaccination or other means, we might inadvertently see a significant surge in these esophageal diseases down the line. We could potentially be trading one set of problems, ulcers, distal gastric cancer, for another, perhaps even more intractable set, esophageal cancer. It's a complex calculation. And this leads to fascinating, albeit still somewhat speculative, implications that might extend even beyond the gut. We've even considered, based on emerging evidence, that the disappearance of H. pylori from human populations, particularly in developed nations over the last century, might also be associated with an increased risk of other emergent diseases that seem to reflect aspects of the current Western lifestyle.
Like the allergies you mentioned.
Exactly. A prime example, as we briefly touched on, is the dramatic increase, the epidemic really, of childhood onset asthma and other allergic disorders seen in industrialized countries over the same period that H. pylori prevalence has been declining. There's that increasing number of epidemiological studies showing an inverse association, less H. pylori, especially Tag A plus strains, correlates with more asthma, hay fever, and other atopic conditions. And critically, as we noted, these associations have been shown to be causal in relevant animal models. H. pylori colonization early in life actually prevented the development of allergic airway disease in mice in those studies. While the exact effect size and the full mechanisms in humans aren't yet definitively established, it paints a profound picture. It suggests a world where our very success in hygiene and medicine, leading to the eradication of a long-standing microbial partner like H. pylori, might inadvertently be leaving our immune systems improperly calibrated, making us more vulnerable to a whole new set of modern health challenges like allergies, perhaps even other autoimmune conditions.
So, what does this all mean for you, the listener, trying to master this complex topic? We've delved deep into the ancient history of H. pylori, explored its intricate biology, how it survives, how it causes damage. We've dissected its complex dance between causing severe diseases like ulcers and stomach cancer, and then, quite surprisingly, potentially offering protection against other conditions like esophageal cancer and maybe even allergies. We've aimed to help you master the clinical approaches used to diagnose and tackle it, understanding the crucial nuances of testing the timing, the different types of tests, and the complexities of multi-drug treatment regimens facing the ever-present and growing challenge of antibiotic resistance. It's not simple.
No, it certainly isn't. This deep dive into Helicobacter pylori, based on Chapter 163, truly underscores how our evolving relationship with ancient microbial partners, organisms we've lived with for eons, might be silently shaping the landscape of modern health challenges in ways we're only just beginning to appreciate. It leaves us with a final thought to ponder. Consider for a moment what other missing microbes, perhaps bacteria or even viruses or fungi that were once ubiquitous in our ancestral environment and on our bodies, might now be absent due to modern lifestyle, sanitation, or medicine. And how might their absence be influencing our well-being, our susceptibility to diseases in ways we haven't even conceived of yet? The story of H. pylori is a powerful reminder that the human body and its microbiome form an incredibly complex, dynamic, and interconnected ecosystem.
It certainly makes you reflect on the delicate balance of our internal ecosystems, and perhaps how little we still truly understand about the roles of many of our microbial co-inhabitants. We hope this deep dive has provided you with the depth of understanding needed to truly master this topic like the professionals in the field, and perhaps encourages you to continue your own journey of discovery, pondering the many invisible forces that shape our health every single day.